Okay, let's dive into this. Welcome back to The Explainer. Today, we are going on an epic, microscopic zoom-in journey down into the absolute building blocks of life itself, the cell. Seriously, everything from the tiniest bacteria to a massive redwood tree, right down to you and me, it's all built from these incredible, microscopic structures. We're going to explore exactly how they work, how they're structured, and the strict rules that govern their existence.
Here's our roadmap for today. We're going to cover seeing the unseen, boundaries and gatekeepers, types of cells, inside the cell city, and finally, growth, rules, and death. Let's get right to it.
Section 1. Seeing the unseen. Overcoming limits.
0.1 millimeters. That is the absolute limit of resolution for the human eye. To put that into perspective, if you draw two tiny dots on a piece of paper and move them closer together than 0.1 millimeters, your eye literally can't tell them apart. They just blur into a single dot. And because a typical cell is way smaller than this limit, it totally makes sense why we desperately needed convex lenses and the magnifying power of microscopes to even realize this whole microscopic world existed.
Let's look at a quick timeline to see how our perspective shifted. Life likely kicked off about 3.5 billion years ago in some pretty extreme environments. Picture boiling hot springs, like the ones in Puga Valley in Ladakh, which house these heat-loving, single-celled bacteria called thermophiles. But here's the wild part. Humans didn't actually lay eyes on a cell until 1665. That's when Robert Hooke used a custom-designed microscope with about 200 to 300 times magnification. Examining a super thin slice of cork, he spotted these little box-like compartments and officially coined the term cells. Fast forward to today's modern era, and we're using powerful electron microscopes. Instead of light, these shoot beams of electrons, letting us reveal structures down to the nanometer scale. That is literally 1/1000th of a meter. Crazy, right?
Moving on to Section 2, Boundaries and Gatekeepers, Crossing Cell Borders.
Now, every single cell out there shares one universal feature, the cell membrane or plasma membrane. The structure of this boundary is incredibly thin. We're talking just 7 to 10 nanometers thick. And it's best explained by the fluid mosaic model, proposed by scientists Singer and Nicholson. It is such a brilliant concept. You see, the membrane isn't just some static brick wall. It's a lipid bilayer, meaning it's made of 2 layers of fat molecules, with proteins embedded all throughout it. Because these molecules can actually move sideways, flip around, and rotate, the whole structure is fluid. And since they're arranged sort of like tiles, we call it a mosaic. Those embedded proteins, they act as literal gatekeepers. They manage diffusion, which is the natural movement of particles from high to low concentration, and osmosis, which is the diffusion of water. They're incredibly picky, selectively letting vital stuff in while blocking the rest.
I absolutely love this analogy for cellular transport. Imagine you're at a friend's wedding. You grab some delicious gulab jamun and you just stuff them directly into your pockets without a bag. I know, it's a messy, hilarious image, but it perfectly illustrates the sometimes messy reality of active and passive cellular transport. Molecules don't always move perfectly and cleanly, you know? They get pushed, pulled, and diffused across that fluid membrane based on concentration and energy, kind of like sticky sweet syrup seeping through fabric. And this brilliantly illustrates a major structural difference when we compare a plant cell to an animal cell. Now, plants can't exactly run away from environmental stresses like wind and rain, right? So just outside their cell membrane, they have this additional, super rigid covering called a cell wall, made mostly of cellulose. It gives them their stiff shape and keeps them standing upright. But animal cells, they completely lack this rigid wall. And that's actually a good thing. Without it, animal cells have the crucial cellular flexibility needed for movement and all the dynamic functions of animal tissues.
Section 3. Types of cells. Cellular architecture. Okay, let's break down the 2 main categories of cells, prokaryotic and eukaryotic. The prefix pro means primitive, and karyon means nucleus. So prokaryotic cells, they have a primitive nucleus, and they're really tiny, usually ranging from just 1 to 10 micrometers. Eukaryotic cells, on the flip side, have a well-defined membrane-bound nucleus, and they are way larger, typically 10 to 100 micrometers. And the other massive architectural difference? Eukaryotic cells are absolutely packed with membrane-bound organelles, whereas prokaryotic cells don't have them at all.
Consider a classic example of a prokaryotic cell, your typical bacteria. Notice how relatively simple its internal structure is. You don't see any of those complex membrane-bound compartments that we just talked about with plant and animal cells. Instead, everything it needs to survive is just happening right there, all sharing the exact same space in the cytoplasm.
The real defining feature here is how they handle their DNA. Because they don't have a true nucleus, prokaryotic cells have to store their genetic blueprints, like their single circular piece of DNA and its accompanying RNA, in a specific region called the nucleoid. Basically, it's just primitive genetic material hanging out in a designated zone, completely lacking any sort of protective, well-defined membrane around it.
All right, Section 4. Inside the cell city, organelle structure.
Let's take a quick tour of the infrastructure inside this microscopic city. The nucleus is basically the central city hall, and it houses the DNA, which is tightly packed together as chromatin. Then you've got the endoplasmic reticulum and the Golgi Apparatus, which handle all the manufacturing, packaging, and shipping. Mitochondria are the essential power plants generating energy, and lysosomes, they're the waste disposal units. Oh, and if we're in a plant cell, you'll also find plastids, like chloroplasts, where a fluid called stroma actually helps capture solar energy. Now, what's really fascinating here is the sheer physical complexity of these structures. The intricate ribbon-like folding of the endoplasmic reticulum, which is split into the smooth ER for making lipids, and the rough ER, which is studded with protein factories, works right alongside the Golgi apparatus. Together, this massive folding creates the huge amount of surface area that's mathematically required to efficiently synthesize, package, and transport materials all over the cell city. It's a highly coordinated logistics network that totally rivals anything humans have built. And we definitely can't forget the tireless workers keeping this city running. Ribosomes. These are the literal tiny protein factories of the cell. They are so incredibly small that under a microscope they usually just look like little dots. You'll find them either floating freely in the cytoplasm or attached directly to the rough endoplasmic reticulum, and they work non-stop to synthesize the proteins the cell needs to function and survive.
Finally, Section 5, Growth, Rules, and Death, the Cellular Life Cycle.
All of these cellular rules are grounded in what we call the classical cell theory. It lays out 3 foundational laws. First, all living organisms are made of one or more cells. Second, the cell is the basic unit of structure and function in all living things. And third, all cells arise from pre-existing cells. It's an elegant concept that beautifully unifies all of biology. It explains the unbroken continuity of life, from the most ancient bacteria all the way up to modern humans. And that continuity relies entirely on the highly precise process of cell division. Under normal, healthy conditions, a process like mitosis safely produces 2 daughter cells that are completely identical to the parent cell, which is how we grow. Meiosis, on the other hand, creates specialized sex cells for reproduction. It's all highly controlled, safe, and absolutely necessary. But it begs a rather terrifying question. What actually happens when these strict cellular rules are broken and the division simply refuses to stop?
So, the crucial control mechanism here is something called contact inhibition. Normally, in animal cells, division naturally shuts down the moment they physically bump into neighboring cells. It's like they know their space is full. But when cells lose this control mechanism, they just keep dividing uncontrollably. And that unrestricted, rule-breaking division is exactly what leads to the formation of tumors and cancer. The loss of just one microscopic rule can collapse the entire cell city.
Now, interestingly enough, cellular death is actually built into the system. It's a feature, not a bug. It's called programmed cell death, or PCD. This is a genetically regulated, highly organized process of selective destruction. It maintains balance and acts as cellular quality control. Amazingly, during human embryonic development, PCD is the exact process that actively eliminates the cells between our developing digits. Think about that. Without programmed cell death, we would literally all be born with webbed hands. Which brings us to a final, pretty profound question. How do cells actually monitor their own growth to maintain this incredibly delicate balance of life? Every single living being, from a microscopic bacterium to you watching this explainer right now, is just a product of these tiny, hard-working, self-regulating cells. It really makes you appreciate the incredible, quiet intelligence of the microscopic world inside you. Thanks so much for joining me on this explainer, and keep questioning the unseen world around you.